Hello friends, welcome to BMH Learning. This video is about 454 sequencing. 454 sequencing uses a large-scale parallel pyrosequencing system capable of sequencing roughly 400 to 600 megabases of DNA per 10-hour run. The process begins with a sample of large double-stranded DNA. The DNA is broken up into fragments of around 400 to 600 base pairs using restriction enzymes that cut the DNA at specific points. Two universal adapters, A and B, are ligated to the ends of the DNA. One adapter, that is adapter B, contains a 5' biotin tag for immobilization of the DNA library onto streptavidin-coated beads. Tiny resin beads are added to the mix. DNA sequences on the beads are complementary to the sequences on the adapters, allowing the DNA fragments to bind directly to the beads, ideally one fragment to each bead. Streptavidin beads are used to isolate only molecules carrying an A and a B adapter. The single-stranded sequencing library is melted from the beads through alkaline treatment. Then, each bead is isolated and amplified using PCR emulsion, which produces about 1 million copies of each DNA fragment on the surface of the bead. The beads are then filtered to remove any that have either failed to attach to any DNA or contain more than one type of DNA fragment. The beads are then transferred to a plate containing many wells called picoteter plate, and the pyrosequencing technique is applied, which involves activation of a series of downstream reactions producing light at each incorporation of nucleotide. By detecting the light emission after each incorporation of nucleotide, the sequence of the DNA fragment is deduced. The use of picoteter plate allows hundreds of thousands of reactions to occur in parallel, considerably increasing sequencing throughput. This was all. Thanks for watching.